Assalamualaikum warahmatullahi wabarakatuh and hello everyone. Today we will present about Sophorolipid application supports biotechnology. This is our team member, our lecturer, Dr. Muhammad Nazrul bin Razwan, group member, Nur Aliha Nandi binti Muhammad Baktia, me Siti Suhaila binti Muhammad Akbar, and lastly Hassan binti Johari. This is our presentation outline. Start with introduction and lastly with the reference. There is a reason why we use biosurfactants Sophorolipid as our biotechnology. This is because it is good in biodegradability and low toxicity, better stability, effectiveness at extreme pH, salinity, and temperature, able to produce from renewable substrates, high production yield and low cost, antifungal properties against plant pathogenic fungus, antimicrobial, antiviral, and immune modulator application. Next problem statement. Fungal infection for tomatoes and chilies causes serious production problems and economically loss. Anthracnose disease on chili was reported reducing marketable yield from 10% to 80% of the chili production in many countries. Other than that, many control of fungus diseases was utilized of synthetic pesticides, which is the chemical or synthetic fungicide brings harmful to the environment and consumer. In recent years, several accidents involving oil spills have been resulting in the environmental pollutions in oceans and shoreline, and the applications of biosurfactants in oil industry are very suitable as a large number of biosurfactants generated are in the application of petroleum related. Biosurfactants are low toxicity and environmentally safe with many advantages compared to synthetic surfactants. As synthetic surfactants that are used are usually hardly degraded by microorganisms, toxic, and also causing environmental damage.
And then moving on to our objective, which is to study the application of sophorolipid as bioinhibition process and antifungal properties, specifically to 1, stimulate the experiment of bioinhibition process for oil spillage in water. And then second, we want to determine the antifungal activity of sophorolipid against tomato fruit rot fungus on Fusarium oldisforum F.sp lycoperdicesi. And then we have methods here, method for sophorolipid as bioinhibition for water treatment in terms of the emulsion index. So the emulsion index E24 is determined according to Cooper and Goldenberg. And then from that, we know that the higher the emulsification index, the higher the emulsification activity of tested biosurfactants. So we can calculate the E24 by using this formula here. The height of the emulsified layer divided by height of the total liquid column, and then we multiply by 100% to get the E24. And then next, we have impact of sophorolipid as bioinhibition for water treatment. And from figure 1, we know that it showed that the graph of emulsification index for hexadecane and cooking oil. And from the graph itself, we can see that cooking oils have the higher value of emulsion index here, which is 67.68% compared to the hexadecane. And the hexadecane here has a constant value of emulsion index, which is 48.78%. So from the graph, we can show that the concentration of sophorolipid increased, the emulsification activity of sophorolipid also increased. So in conclusion, this shows that sophorolipid can emulsify cooking oil, thus enhancing the water solubility, decreasing surface tension, and increasing the oil degradation. So next, we have figure 2 here. Figure 2 is a bar graph of a comparison for cooking oil after bioinhibition process. So based on figure 2, we assume that the weight of oil for the control treatment on water and oils only, with the absence of sophorolipid, is remain unchanged. So meanwhile, the weight of oil with the presence of sophorolipid is decreased. The analysis shows the oil in the control treatment showed no oil biodegradation, while high level of biodegradation were observed in the treatment with sophorolipids.
Okay, next we move to the second application of sophorolipid, in which for antifungal activity against tomato fruit rot fungus. The methods will be divided into two, which first one is poison agar test. Firstly, we need to obtain the pure tomato fruit rot fungal from the Department of Biology Faculty Science CPM. Second, we did the cultivation of pure fungal Fusarium oxysporum FSP, Lenco persicci for 7 days. Next one, we prepared the poison agar plate, and finally, we conducted the poison agar experiment and measured the growth of fungal mycelium after 7 days. For the second division, which is the surface coating, firstly, we prepared the different concentration of sophorolipid, in which for this experiment, we made maximum 5%. Next, the second step, there will be one sample uncoated, while another two samples will be coated with different concentration of sophorolipid and we will keep it under room temperature. Finally, we did the observation for the condition of the samples. So we move to the results for the poison agar test first. As we can see in the figure 3, it shows the graph of relationship in inhibition of mycelial growth versus the treatment of concentration. So we could see clearly that whenever the concentration increased, the inhibition of mycelial growth also increased. For the maximum concentration, which is 5% sophorolipid, it recorded the highest inhibition of mycelial growth, in which 76%. So it shows that sophorolipid could give positive effect of the inhibition growth of the fungal pathogen. So this is the results for the surface coating. So we have the uncoated tomato and also the coated tomato with 5% sophorolipid. So we could see the result for the after the observation. So we could see that the uncoated tomato showed more visible spoilage on the surface, while for the coated tomato with 5% sophorolipid, it shows less sign of visible spoilage on the surface. So it's shown that sophorolipid is able to prolong the shelf life of the tomato fruit. So we have come up with the conclusion. So for the first application of sophorolipid, it shows that emulsification activity demonstrated by the cooking oils shown that it has a great potential in bioremediation, and also the application of sophorolipids in bioremediation could be a potential alternative as their synthetic surfactant in order to treat the oil pollution, especially in the ocean or the water bodies. And also the effectiveness of biosurfactants in bioremediation is expected to be extremely significant in the future. For the second application, it shows that sophorolipid gives positive effect on the inhibition growth of the mycelium of the fungal pathogen. And also it could be potentially as biopesticides or also biofungicide. And finally, sophorolipid is able to prolong the shelf life of the tomato fruit.